think, oops, there we go. So like I said, I'm Karen. I'm a research scientist at Bigelow Laboratory for Ocean Sciences. I'm going to talk to you about mixotrophs in the North Atlantic Continuous Plankton Recorder time series. This is a project that started quite a few years ago and actually, I mean, nearly reached completion quite a few years ago as well. But I wanted to give a shout out to the Eco Days program because this paper or this work that I'm going to present is directly a result from this symposium that brings together early career researchers from across the aquatic sciences. It's funded by NSF and ASLO. And I will say that I think it's 4 or 5 of us in this working group all met at this Eco Days in 2014, something like that. Anyways, I just wanted to show this advertisement, or it's not advertisement, but description to you because your students or your postdocs 2016, thanks Jess, may be interested in this program. It's a great program and it happens, I think, every other year. So yay, Eco Days. And like I said, this project is essentially a paper at this point. It was co-authored by myself, Jessica Liu, and Nicole Millette, who are all co-leads of this working group. So this working group was another product of that Eco Days session. And also a few other people contributed as well. But formally, this is a talk about large protistan mixotrophs in the continuous plankton recorder time series, their patterns over the last 55 years as it relates to biogeography and seasonality and environmental conditions. Our paper starts out basically introducing Stucker's hypothesis that she posed that mixotrophs will dominate in a primary producer community when either light or nutrient supply is limiting. And so we took this and wanted to test it on a large scale, both large spatial and long temporal scale. So we decided to use data from the Continuous Plankton Recorder time series, or CPR time series. There are some shortcomings of this set, especially when it comes to phytoplankton. I shouldn't really lead with that, but I do want to acknowledge off the bat that we're looking at large protists that will be cast as CPR is usually you can actually look at zooplankton. So we're talking about the larger of that this group has been discussed. This map shows all of the continuous plankton recorders that were used, and then the blue and yellow indicates what we consider shelf regions, or the yellow ones, and then the blue offshore regions. So
Karen,
metrics
this is Heather.
Considered to look at this question of mixotrophy. Yep.
Can you hear me?
Hi, Heather.
You're breaking up a little bit. Is she breaking up for others as well?
I can hear you. Can you hear me?
Yes, I hear the same thing. It's breaking
up for me too.
You might want to just turn off your video. That might help.
All right, I'll do that. I don't have an Ethernet cable plug with me. I would do that. Let's see. Sorry, I'm gonna have to. Oops. Give me 1 second here. Trying to access this thing. Trying to turn my video.
Stop your video, Karen.
What's that? You can do it?
Stop your video.
Why I can't
There you
find
go.
it. Thanks. All right, hopefully that'll be better.
And just make your slides full screen again. Perfect.
Okay, sorry about that. So yeah, we're looking in this data set at the proportions of autotrophs, heterotrophs, and mixotrophs in that large protistan community. And then we use those relative proportions to create what we are calling a trophic index. So a number that goes from -1 to +1, where -1 represents 100% autotrophs in that community, and +1 would be 100% heterotrophs. 0 would be 0 tends towards mixotrophs. However, I will say if you have 50% autotrophs and 50% heterotrophs, you would also get a 0. But I'll explain why it works out fine anyway in a second. So our spatial scales that we looked at were CPR, or continuous plankton recorder regions, which have been used to look at they've been used in other studies, and they're delineated by oceanographic features. We also turn the data into a 1-degree grid for finer resolution, spatial resolution. We looked at the data on seasonal and annual timescales, and we used environmental climatologies to see what environmental factors were driving the trophic index, as well as the proportion of mixotrophs in the community. So the first thing we did was to see which continuous plankton recorder regions co-varied in terms of their seasonality of the trophic index. And this is where we saw that we needed to separate offshore regions from shelf regions. So this just showed us that in terms of biogeography, the shelf regions were varying over seasons similarly, and then the offshore regions were also varying similarly for the most part. Okay, so again, we divided shelf and offshore. So on the left-hand plot, we're looking at data for shelf organisms, and then offshore on the right. On the left-hand side of both plots, it's the relative proportion of autotrophs, heterotrophs, or mixotrophs from a scale of, it would be 0 to 1, but they never were that high. So 0 to 0.8. On the right, we have our trophic index, and that's the red axis and the red line. And so what you can see is the seasonality of these different organisms and what's driving our trophic index. So when I mentioned that 0 in the trophic index really indicates mixotrophs being highly present, it's because our heterotrophs are generally very low in this data set. So that's this dashed line. So you can think of the trophic index as an indicator of basically when you have more autotrophs versus more mixotrophs. And then you can see that the seasonality of mixotrophs and the trophic index is different slightly for shelf versus offshore regions, with the trophic index increasing earlier or staying high. Sorry, it's just slightly different. It's yeah, it just goes lower in the shelf regions. And so yeah. The spatial results I think are really neat. So this is the trophic index for winter here in the upper left-hand corner, spring below that, summer, and autumn. And so the dark blue colors represent community samples that are more autotrophic, or sorry, have more autotrophs in them. And then the gray ones, the grayer areas are more mixotrophs, have more mixotrophs, and then the sporadic little red points are where we had a dominance of heterotrophs occasionally. And so you can see that as you move into the summer region, you have more mixotrophs and autotrophs. Sorry, the summer season, you have more of these mixotrophs and autotrophs moving further north. And in autumn, they're dominating in the central part of the study area, and generally in the sort of western and northern edge, we have more autotrophs most of the year. So getting back to Diane Sucker's hypothesis, we looked at the relationship between environmental variables, specifically nitrate and light, or PAR, to the mixotroph proportion in the communities. And for this, we used the continuous plankton recorder regions, and we did, each point represents a region and a month. So this is averaged by, or this is by every month in the time series. Was that a sneeze or a comment? Or neither? Okay. So the warmer colors or the yellower colors represent more mixotrophs in that sample, and then the bluer colors we have more. Well, not not as many. In this case, it's more autotrophs. And so what you can see is that when nitrate is really low, we have more mixotrophs that are on a range of light levels, but the higher the light in this low nutrient condition, the more mixotrophs we have in that community. This is for offshore, and then a similar pattern appears for shelf regions. And then likewise, when we have really low light levels, when we get to these higher nitrate levels, we have slightly more autotrophs, although the pattern is, sorry, mixotrophs, oops, although the pattern's not as distinct. So I would say this confirms Stucker's hypothesis to some degree. I don't want to harp on the modeling too much. I'm kind of loath to show you a table in a presentation, but I just want to point out we did model this using our environmental parameters, using both spatial resolutions that we looked at, both the 1 degree and the CPR regions. And I just wanted to point out one thing that stuck out to me and sort of put it out to the group as an interesting point, which is when we considered the higher resolution spatial data, the shelf mixotrophy was predicted more when silicate was included in the model versus phosphate in offshore regions. And I think this could be kind of an interesting indication of the difference in driving factors for phytoplankton community composition, whether it's impacting mixotrophs directly or other organisms that can outcompete mixotrophs under certain conditions. I just think that was an interesting difference. When we go to a coarser spatial resolution, the CPR regions, the models are very similar and include light, temperature, and nutrients. I should say we developed these models using a stepwise linear regression method that gives us the simplest model that has the strongest predictive capability. So lastly, I guess this is my last slide. Lastly, I want to show you the interannual patterns of these different regions. So just to remind you again, we have shelf regions that are in yellow there, and then offshore regions in blue that are showing a really different pattern when it comes to changes over the time series. So the time series we considered for this was from 1958 to 2015. So to orient you, the left-hand plot is for the shelf regions, the right hand is the offshore regions, and the x-axis is months, so we're going from January to December, and the y-axis is years. And so what you can see for the shelf is, I mean, kind of an interesting change potentially. Oh, sorry, I should also mention this is the anomaly of the trophic index. This isn't just the pure trophic index itself, but the anomaly from the whole average of the time series. What you can see here is there's not really a super clear trend in change in the abundance of mixotrophs over the whole time series, but there may be an interesting change in phenology. So we get this, basically the pattern was that you have lower trophic index or more autotrophs in the winter and spring, moving into more mixotrophs in the summer and fall. But then as you get into the more contemporary time period, you see that shift to the opposite. And so I think that's really interesting. And it's quite different from the pattern we see offshore, where in general, there's just an overall increase in that trophic index or a tendency towards more mixotrophs over autotrophs. So that is the last slide I have to show you here. And I guess, I wouldn't mind taking a couple questions since I went pretty quickly. If anyone has questions, let me stop sharing my screen. Does anyone have any specific questions about what I presented? Okay, well, that's great. So now we'll have Kyle give his talk. I'll introduce him. Let me just pull up. I'll tell you who Kyle is if I can find my window here. All right, so Kyle is an associate professor at University of Hawaii in Manoa. So thank you for joining us pretty early in the morning, although I understand you've already given a lecture this morning, so you should be all warmed up. He studies the ecology of phytoplankton and planktonic and benthic communities in general, and is interested in the maintenance of species diversity, scaling from community interactions to ecosystem processes, and looks at things through ecosystems through a trait-based lens, and uses trait-based approaches to study community structure. He's also interested in theoretical ecology and synthesis of models with empirical research. So yeah, Kyle, thank you for speaking to us. I will hand it over to you if you want to share your screen and go ahead.
Great. Thanks a lot, Karen. I did actually have a question for you. Can I ask it now? Or do you
Yeah,
want
of
me to
course.
talk
Yeah.
about it later?
No, it's fine. If it's specific to my talk, go ahead.
Yeah, so you were showing that interesting plot where it seemed like the relationship between light and mixotrophs switched depending upon the nutrient concentration.
Yeah.
But then when you're showing your like table of the best models, it seemed like there was not an interaction between nutrients and light in the model. So do you know what I'm getting at there?
Yeah, I do. So I actually I picked models that didn't include interactions specifically because it was confusing.
Yeah.
What I found strange about the models was that there was basically they have a negative relation. It shows the best model was a negative relationship with light and a negative relationship with nutrient or with nitrate. And I would expect there to be one positive and one negative based on that plot. But I think I could go back and run the modeling again with interactions. I do believe there was a model that had interaction between a lot of different terms, including light and nitrate. I was just
Yeah.
I have trouble interpreting those sometimes.
Yeah, no, it's especially challenging if you're doing like a stepwise approach and including like all possible interactions. It's going to get fairly confusing, but yeah, anyways, yeah, that was my question. So I can go ahead and start.
Yeah, go ahead. Thanks for your question. I will go back and look at it and see if I can simplify it so that I can get at that interaction because I think it's a pretty clear signal in the data. So
It looked pretty interesting from the plot, so it would be good to put it in the model, I think. Yeah, or see, at least see how much it affects the model.
yeah, thanks.
Now I'm trying to get this to let me share.
Okay, Heather or May, is there a trick to?
I think the problem is I haven't used this app before. Sorry, I'm gonna need to like.
It might require you to log out and go back in again.
Yeah, I'm gonna need to quit and be back in 1 second.
Oh, that's
so
okay.
so
I did
sorry
the
about
same
that.
thing earlier. That's okay. Don't worry about it. Take your time.
Any more questions for Karen while we're waiting?
Yes, Lucy.
Yeah, thanks a lot, Karen. I have a question. So you are calculating the global index on the entire community. And so did you try to also have a look, because it's a global index telling you that there is more autotroph or there is more mixotroph. But did you have a look in detail, in fact? I mean, is it really dominating entirely by autotrophs, or can you give us insight? I mean, what is the proportion of guys that there are behind, in fact?
Right, yeah. So I tried to show that a little bit in the seasonal plot, showing just the climatologies of the proportion of autotrophs, heterotrophs, and mixotrophs. And the highest that the mixotroph proportion gets is about 70% of the sample or the community at that point. And the highest the autotrophs get is close to 80%. So it does, yeah, it's not getting to like 0 and 1 or 0 and 100%, but those proportions do get quite high and quite low throughout the seasons.
All right. And just to know, to do this assignation, I mean, to autotrophs or mixotrophs or heterotrophs, which kind of database are you using? You have your own database, or you're using, I don't know, something like so the publication, for instance, from Schneider et al., which is quite recent. You
Yeah,
have that.
we actually went through by hand ourselves and looked it up and assigned the trophic strategy for each species in the data set ourselves.
Mm-hmm.
And that was
Right.
just from the literature.
And so you have these 3 classes, and you do not intend, I mean, to use I don't know, the classes from Mitra et al., for instance, with different mixotypes, you know,
Yeah.
to
We actually didn't do that because when we did this analysis, I don't think that those publications or that information had been generated yet. We actually did this quite a while ago.
okay, but okay.
But we
Thank you.
could do that now.
Yeah,
Yeah.
no, just to you know, to have maybe you know, like a finer picture, I would say, a bit, because the 3 classes, it's already certainly giving you very nice trends, and so I was wondering, yeah. Okay, but
Yeah,
thanks a lot.
Yeah, that's a great idea. Thank you. All right, let's
Bye.
turn it over now to Kyle, who hopefully can share his screen.
Yeah, so can you see my screen?
Yes.
Okay, and are you
I don't
seeing?
I can see it, but it's small. It might just be on my end.
I mean,
Yeah, I think if you hit presenter view, it will maybe be better.
I see. Okay.
Great.
Wasn't working. It's slightly different than Zoom, sorry. Okay. So it looks good now?
Yes.
Okay. All right. Thanks for having me. For those who saw my talk Ocean Sciences, this is gonna be very similar. So apologies for the repetition. So what we're interested in here is thinking about trophic strategies among small eukaryotic phytoplankton and how they relate to the sort of diversity and biogeographic patterns of those organisms. Just wanted to acknowledge my collaborators down here. Greg Stewart is the co-PI on this work, and the data come from a ton of work by Chin Lee, Chris Schwartz, and Kelsey McBain, a lot of experiments. So we're thinking about the smallest eukaryotic phytoplankton here, and by small, I'm saying roughly less than 5 microns in size. We know they're really important in the open ocean. They can rival cyanobacteria in terms of contributions to productivity. People have shown that they're the major predators of bacteria, so phagotrophic mixotrophs, at least some of them are phagotrophic mixotrophs. And one of the things that's been interesting to me for a long time is just how diverse this sort of small size class is. There's all these different classes and higher taxonomic groups of eukaryotes. And, you know, I would say we don't have a great handle on what's different about them. You know, how are they functionally different? How is their ecology different? In part because they're hard to tell apart using microscopy or flow cytometry. And so they've maybe gotten less attention than larger phytoplankton or than things like Prochlorococcus and Snectococcus, which you can count with the flow cytometer. So we're really just trying to think about, does all that phylogenetic diversity translate into functional diversity? How many of these different groups are mixotrophs? What are the trade-offs that affect, when is it good to be a mixotroph? And like, what do the patterns of community structure look like? So Chris Schwartz, who I mentioned before, isolated a huge amount of strains from Station Aloha in the North Pacific subtropical gyre. And don't have time to get into them in a lot of detail, but they're all in this 2 to 5 micron size range. There's 39 of them that we showed will eat and grow mixotrophically on Prochlorococcus as prey. They're from 6 different taxonomic classes, including a number of clades that had not been isolated before. So including a number of different kinds of dictyocophytes, and then bolidophytes, chrysophytes, haptophytes, chlorarachnophytes, and then we've got one dinoflagellate in the mix here. So these are all mixotrophs. Some of them were known to be mixotrophs before. A lot of them were suspected, but this is the first time they've been shown to be mixotrophs and shown to consume Prochlorococcus. And then if we look at their sort of functional capacity, one thing that's really interesting is that they vary a lot in how quickly they graze. So this is the specific clearance rate, the amount of volume of water cleared of Prochlorococcus per grazer by body volume per time. And that tends to be highest among the chrysophytes that we isolated, lowest amongst this genus Florentiella dictyocophytes, and then there's a whole kind of spectrum in between. And one of the things we found in these grazing experiments is that the strains that graze faster tend to have a lower apparent conversion efficiency. So, the amount of grazer biovolume created per prey biovolume consumed. So it suggests that there's, kind of a spectrum here where maybe you have faster grazers that are respiring more of their prey biomass, doing less phototrophy. And you have slower grazers that are doing more phototrophy and creating more biomass per prey consumed. And then we wanted to dig more into this, sort of potential spectrum and trade-offs between phototrophy and phagotrophy among this diverse group of organisms. And so we did some experiments looking at growth rates under either low light or high light. And then in addition to all those diverse mixotrophs I mentioned, we also added in some autotrophs. So these were some prasinophytes and one plagophyte, Plagomonas. None of these appear to be able to eat Prochlorococcus based on our experiments. So we're calling them autotrophs. And if we contrast them with the mixotrophs, we find that under low light, which is around 10 micromoles photons per meter squared per second, the autotrophs can all grow fine. And that's about 1% of the surface irradiance of Station Aloha. They don't grow super fast, but they grow fine. The mixotrophs, only one of them can grow autotrophically under those conditions. So we're not feeding them this experiment. We're trying to tease apart these different functions here. And then if we look at how fast they grew under higher irradiance, all of them could grow except for the chrysophyte in this experiment. That one cannot grow autotrophically. It needs to be fed. The rest of them can grow, but there seems to be a correlation such that the ones that are the faster grazers grow more slowly under these autotrophic conditions. So that's again consistent with there being this spectrum of trophic strategies from obligate mixotrophs to things like these dictyocophytes over here that can grow as fast as the autotrophs under autotrophic conditions, but don't graze very fast. Oh yeah, that's what I just said. Of course, it'd be interesting to know more about what the mechanism of what's going on here. We don't know that at this point, but it could be driven by respiratory costs or investment in photosynthesis or both. And then the next step was we wanted to sort of, link this trait diversity, and autotrophy, and the spectrum of mixotrophs to where they live in the ocean to try to test these kind of hypotheses that Karen was talking about. And so to do that, we basically matched up the strains we'd isolated with data from the Terra Oceans project. So they have this 18S V9 region ribosomal RNA metabarcode data set, just helpfully size fractionated. So we're focusing on this 0.8 to 5 micron size fraction. And these are the locations where they have that data from. So they're mostly from the open ocean and mostly more kind of oligotrophic to mesotrophic conditions, which is where these small phytoplankton are the most important. And then, well, I'm not going to show it, but basically we use the 18S sequence data from our isolates to match them to the OTUs in the Terra Ocean data set. Okay, so when we start looking at niches, this is just a quick view of like the more raw, low-level data of depth niches. So there were samples at surface, which is 3 to 7 meters, and then at the deep chlorophyll maximum at all of these stations. And one thing that pops out is that all of the autotrophs are relatively more abundant at the DCM compared to the surface. So Ostriococcus, DCM versus surface. Chloropecon, that's percentophyte, DCM versus surface. And then there's only one mixotroph that's like that, one of these Florenciella OTUs. And then the rest of the mixotrophs are all more abundant at the surface compared to at the DCM. And then if we look at that spectrum of grazing abilities that I showed before, there's a correlation such that the mixotrophs that graze faster have shallower distributions. So that's consistent with just comparing mixotrophs or autotrophs, or if you look within the mixotrophs, you see the same pattern arising. So this on the y-axis here, this is the ratio of relative abundance in the surface versus the DCM. So if it's above 1, more abundant in the surface, below 1, more abundant at the DCM. And then this is this grazing rate on the x-axis. Okay, so that's what's going on across depths, and then we wanted to look across all these locations of how the environment's varying. And so one way to look at that is to do like an ordination to see what are the major axes in community composition. So focusing on these OTUs, which are a large fraction of the phytoplankton community in this size fraction. About 46% of the variation among those OTUs can be explained by one principal coordinate axis. And along that axis, you've got, like, in this direction, higher nitrate, higher chlorophyll A concentrations, deeper mixed layers, and in the other direction, you have higher temperatures, and then this is par at the sample depth. So this basically looks like axis of stratification, deeper mixing, more nutrients, less light, cooler temperatures, higher chlorophyll, and vice versa. So that's the major axis in community structure, seems to be correlated with stratification, which is going to drive nutrient supply. And then if you look at how the different OTUs fall out along this principal coordinate axis, there's again a correlation between how quickly they graze in our lab experiments and what their principal coordinate score is. So the slower grazers, here's the autotrophs, and here's the more slowly grazing mixotrophs. They're more associated with these more deeply mixed, higher chlorophyll A conditions, and then the faster grazers with these more stratified conditions. You see a similar pattern. It's statistically a bit stronger if you just look at chlorophyll A. So this is just the slope. If you do a generalized linear mixed model, look at the slope of relative abundance versus surface chlorophyll concentrations. The autotrophs tend to increase with chlorophyll A, and then there's this one more autotrophic mixotroph that does, and then the really fast grazing mixotrophs more strongly decline as chlorophyll A increases. So this is kind of the conceptual diagram of what we think is going on here. We've got two parallel gradients across, these are major physical gradients in the ocean, and we're seeing parallel patterns in community structure along this trophic spectrum across these gradients. So if you go from, less stratified to more stratified, you go from more phototrophic phytoplankton to more phagotrophic phytoplankton. And if you look across depths, again, at deeper depths, you have more phototrophic phytoplankton, shallower depths, more phagotrophic. And then this panel on the right here is just showing how these different taxa fall out along this trophic spectrum that we have some evidence for. So these are the autotrophic taxa down here, and then this is going up from more autotrophic to more mixotrophic or heterotrophic mixotrophs. And the size of these circles is just showing their average relative abundance in the Terra Oceans data set. Okay, so these are my conclusions. We've got a phylogenetically diverse spectrum here of phytoplankton, including a wide diversity of mixotrophs. Differences in their niches are strongly correlated with where they lie in this trophic spectrum. It seems like nutrient supply and maybe light are the likely drivers. We did some modeling of this, which I didn't get into because it would have taken too long, but could talk about that more in the discussion. Some of the interesting implications, I guess connecting back to what Karen was talking about earlier, the relationship with stratification here, because the oceans are getting more stratified and will continue to do so, it implies that the future oceans are going to be more mixotrophic based on our data as well. And one thing I'm interested in is, what are the sort of ecosystem and food web consequences of these shifts in community structure? It seems like there should be predictable shifts, and it would be interesting to talk more about what those should be and how they could be measured. And it also seems like we might be able to better correlate trophic strategy of the phytoplankton with things you can sense from space, for example, if we have a strong correlation with chlorophyll concentration. Okay, thanks for your attention. That's all I have for today.
Thanks, Kyle. That was really nice. I guess I'm always impressed when a really coarse scale analysis like I presented, and the results of that correspond to the results of a really fine scale detailed analysis like yours. So that's kind of neat. Does anyone have any questions for Kyle specific to his talk before we get into the discussion questions? I actually can't see if anyone's raising their hand, so you should just go ahead and speak up if you're raising your hand. Oh, I see, actually, sorry. Jessica, you're raising your hand, I think.
Oh, I think Lucy was first.
Oh, Lucy, please go ahead.
Okay, all right. Thanks. Thanks, Kyle. So thanks a lot. It's really interesting. And so I was wondering, as you have in fact focused on the smallest size fraction, what would be your expectation for the biggest one? Because I guess it's mainly constitutive mixotrophs, most of them in the small size fraction. So I know a bit the Terra Ocean dataset, and I know there are differences, by size fraction. So what would be your expectation for the biggest one? Yeah.
Generally speaking, I would expect it to look similar, regardless of the size fraction, just because I don't, in my mind, the sort of competitive mechanisms that are driving when it's good to be a mixotroph are the same across size classes. It's just that you might see like some shift in, the fraction, like the overall fraction of mixotrophs across size fractions. Like Ken Anderson's group has done some nice modeling, sort of explaining why that might be, just because larger phytoplankton are going to be more strongly affected by nutrient limitation due to diffusive boundary layer. But in terms of the major patterns, though, I would expect them to be similar. As far as constitutive mixotrophs versus non-constitutive mixotrophs, I haven't thought about that as much. I'd be interested to hear other people's thoughts on that. It seems like, the constitutive mixotrophs potentially might be more strongly constrained by the presence of the organisms that they're stealing plastids from, or
Sure.
yeah.
Because okay, my guess would be maybe I don't know, I would see the small one would be rather constitutive mixotroph, and for the biggest one like radiolaria and so on, which are doing symbiosis or dino, or maybe for some of them I would expect maybe because they have a different biogeography. I mean, and for the lineage that you have shown, it's mainly so haptophytes, and I was not able to to read them all quickly for your tree.
Yeah, no, these are all
But,
constitutive mixotrophs, the ones that sorry, I can't figure out how to unshare over here, but yeah, the taxa that we were looking at are all constitutive mixotrophs. So yeah, that's correct. Your supposition is correct.
okay, but no, okay, no, it's okay. But thanks. I mean, for the small, it's really great.
Jessica, do you want to ask a question?
Yeah, Kyle, this is very interesting. I was wondering about the observation that you made about phagotrophs being higher in the water column, and the potential for UV damage. Do you think that? Well, first, were you able to find genes that here from the Terra Oceans data set that you know of that protect against UV, and would that sensitivity scale maybe with the number of chloroplasts?
Yeah, it's an interesting question. Yeah, we haven't done any genomics in this as part of this analysis, so it's all based on metabarcoding. So just taxonomy, like who's there and who's not. Would it scale? Yeah, I mean the more like photon capturing machinery you have, then the more damaging it's going to be if you're getting too many photons, right? So like that makes sense to me. So that could be another interesting factor sort of on top of the others. There's a lot of things that co-vary, of course, with depth and with stratification, nutrients and light, and too much light versus not enough light. So
Yeah.
and temperature could be playing a role as well. I kind of expect it's not the major driver, but it could also be driving higher phagotrophy as well. So it's possible there's multiple sort of things going on at the same time, reinforcing the pattern, which is why it's so strong.
Yeah. Cool. Thank you. Very interesting.
I think we have time really for one more question, because we need to get to our discussion. Sorry, Holly. I'm sorry if I'm mispronouncing your name, but I think you had your hand up next.
You did a wonderful job with my name.
Oh, good.
The question is, is there a difference in the oxygen tension, and therefore in terms of the distribution of the mixotrophs? And also, a corollary to that is, what happens with oxygen toxicity? If there's too much light, you're going to have too much photosynthesis, too many oxygen radicals, and you're going to start getting a negative impact. So have you considered that in your analyses?
No, but thanks for that comment. I hadn't thought about that at all, but now I'll have to think about that some more. Yeah.
Okay, great.
Yeah.
Thanks for all the great questions. Holly, I'm really sorry to cut you off from your question, but I really want to get to our discussion questions because Kyle, Jessica, and myself have got some good discussion questions for everybody. But maybe at the end, there might be time for your question, Holly. I hate to cut people off. Okay, so I'm gonna put a link into the chat here for the discussion questions, and just to remind people how this works. I'm gonna make sure we can everyone can view this. So everyone can sign on Yep, so anyone with the link. Okay, great. So everyone should be able to sign onto that link and see the discussion questions. There are quite a few of them, and I think what we'll do is break into, let's see, maybe 4 groups. Does that sound? I think Nicole was going to set up the groups.
There's 4 groups already set up.
Oh, amazing. Okay, you guys are the best. So, you'll be assigned into a breakout room, discussion room. If you can self-select a kind of a person who will be in charge of reporting out, so maybe taking some notes and reporting out from your group's discussion. And like I said, there are quite a few, so if you want to select certain ones to discuss, that's great. Or you can just start at the top and work your way through. I think we'll reconvene at, let's say, maybe let's do 20 of 5. That will give everyone 20 minutes to discuss as a breakout group and then 20 minutes back together. Does that sound good, Nicole? Millette?
Yeah. Yeah, that sounds good.
Okay, great. So self-select someone to report out and enjoy your breakout discussions. Thanks so much for the talk, Kyle. That was great. Always such an abrupt change back to the whole group all of a sudden. I'm going to just give everyone a moment to finish writing their notes. Mainly, I'm giving myself a moment to finish writing the notes. All right. So, okay. Thanks for everybody who's stuck around so far. We had some great discussion. Why don't we just go through each group and maybe, so group one, maybe your person can report out which questions you looked at and how your discussion went.
Great, thanks. Hi, everybody. I'm Ashley. I'm reporting for group one, which was Stephanie, Kevin, Nicole, Millette, and myself. We really liked question number 4. We're really excited about the idea of instituting time series data efforts and where to do that. And so all we did right now on the document is list out all the options, existing options there are right now. So Karen talked about CPR. There's also Aloha, BATS, this Martha's Vineyard Coastal Observatory, the North Atlantic Bloom Experiment, Palmer, NOAA AMLR, Chesapeake Bay Program, SPOT, Santa Barbara has something in the works, and then there's some quarterly cruises coming out of Woods Hole. So we just listed out everything we could think of. One thing that we didn't know was more international data sets, Europe, South Korea, Japan, China, places that might have some long-term stuff going on. Not time series, but it would be awesome to jump onto other campaigns that aren't necessarily in one spot, that go through gradients, so you can compare mixotrophy over gradients. So the things that came to mind during our breakout were Ghost Ship, Geotraces, Tara, Simon's gradients and scope, and the AMT, the Atlantic Transects. And then finally, we really had fun talking about our dream. What do we really want in a perfect world of unlimited resources? We would have occupations from pole to pole in the Pacific, in the Atlantic, and from tropics to pole in the Indian Ocean, where we were equipped to look at mixotrophy at all angles. So that was what we talked about.
Awesome. Sounds great. So group 2, that was myself, Matt Johnson, Suzanne Wilkin, and where are you? Oh, Natalie Cohen. So we started out with question 2. So we were talking about the distribution of mixotrophs versus mixotrophy. And then we just riffed from there and went off into just mixotrophs outer space. But so we thought that there would be similar distributions. You can't really decouple mixotrophs from mixotrophy, and because it's so relatively costly, if you're not using that potential, then you're probably at a disadvantage. So in general, there should be a good correlation between the biogeography of mixotrophs and mixotrophy. But we also got into discussing when mixotrophs, I guess this plays into question number, question about ecosystem function. When mixotrophs might stabilize the system. So in systems where there's sort of high amplitude swings in environmental variables, so things not like the oligotrophic ocean, but maybe more subpolar, polar, or even temperate places where you have seasonality, mixotrophs being flexible might use more mixotrophy in those places during times of transition, and possibly stabilize those systems in terms of primary production. We also talked about coastal systems, that specifically, some coastal systems are macronutrient limited, but there's plenty of light. Some have eutrophication. So we started out talking about coastal systems very generally, and then realized that there are lots of different coastal systems, and kind of decided that we just need more observations of I think this group talks about this a lot. We need more observations of mixotrophy, because based on some of the larger analyses, like Suzanne Olella's analysis, it seems that mixotrophs are everywhere almost all the time. but when they're using mixotrophy is still a big mystery. Let's see, what else did we talk about? We talked about comparing systems. So in terms of biogeography, comparing things like oligotrophic versus Arctic systems, especially with regard, well, we were talking specifically about depth and the depth distribution of mixotrophs and mixotrophy, that's a really interesting scale or axis to look at that isn't often looked at, but it's very challenging to compare between systems. Although once you do tease apart the environmental factors that are giving you the depth or spatial distributions of mixotrophs and mixotrophy in different systems, those comparisons are where you really start to get into the mechanisms behind that biogeography. We also talked about the challenges of studying what mixotrophs are eating. And so just understanding how the distribution of prey impacts mixotrophs and then they're using mixotrophy. I think if anyone from my group has anything to add, please do. Otherwise, we can go on to group 3.
Hi, so I'm supposed to represent group 3, but I desperately need to take a phone call, so I do have to sign off. Would somebody else mind taking it? I'm really sorry about this.
No problem. It happened. Anyone, or we can go to group 4 and come back to group 3 so someone else can organize their thoughts. Let's do that. Group 4, do you want to go ahead?
Sure. I guess, actually, does anybody from my group want to discuss our notes? Okay. I'll do it. I took the notes. I'm not as good of a note taker as some of the other co-conveners of this working group, but anyway, so we started talking about mixotrophy distributions. Our group was myself, Kyle, Sophie, and Isat, and started talking about the effect of light, about how talk from Karen and talk from Kyle kind of had contrasting effects of light on mixotrophy, that low light might select for mixotrophy if you're comparing against autotrophy, but then that trade-off would flip if comparing against heterotrophy. We talked a little bit about the distinction between oceans versus freshwater systems, and a little bit more about photo damage, and how Kyle had some qPCR data that showing lower absolute abundance close to the surface, but still greater relative abundance of mixotrophs compared to autotrophs. We kept talking a little bit about nutrients, actual distributions of nitrate at surface and by depth, and the effect of taking up dissolved organics. Shifting to question number 2, we didn't really have an answer for the distinction between the distributions of mixotrophs versus mixotrophy. And I suggested that maybe in very competitive environments, mixotrophs may not be there unless they were actually engaging in mixotrophy. Otherwise, they would not survive. And again, that maybe in simpler systems, like different lake systems, it'd be easier to look at these trade-offs. Regarding the difference in ecosystem function due to mixotrophs, we talked about how we may actually test the predictions from models that mixotrophs within the system would result in greater retention of nutrients in the food chain versus remineralization, and talked about different types of maybe experimental setups, such as looking at gross growth efficiency for mixotrophs versus heterotrophs under different nutrient light conditions, But then we started talking about the effect of oxygen before we got cut off, and I didn't catch the rest of it. Is that it? Did I miss something? To my group.
Good work. Thank you.
Good summary.
Thanks.
All right, do we have, I think, Holly, are you gonna speak for group 3?
Sure, yeah. I think we covered some of the same ground as the other groups, so I'll try to be brief. Essentially, I started by saying that I wanted to know whether we could link Mixotroph Biogeography to Mixotrophy Biogeography, and very quickly we talked about how one of the main challenges of that is that we have very limited data sets with which to do such comparisons or try to get at the link between mixotroph distribution and their biogeochemical function distribution, because most of our data sets are focused on presence absence either of organisms or of particular genes, although we're building data on mixotroph function and activity. We also talked a bit about how one would attempt to measure nutrient cycling and other elements of mixotroph activity to try to link those with mixotroph abundance. And we talked about some possible tools that could be used, including some statistical approaches like correlation matrices, especially as we're in an age of a real wealth of omics data, looking for correlations between organisms as well as correlations between types of organisms and their environments might give us some really useful context to link the presence of organisms with what they might be doing in a particular location. And we also talked about the possibility of combining empirical data and relatively high information mechanistic data on performance of cells in terms of uptake rates and photosynthetic rates that we might have from the laboratory, with mathematical models, with environmental data about the distribution of resources that mixotrophs use, and asking models what sort of optimal mixotroph or optimal mixotrophic activity they might predict, and contrasting that with some really focused field studies on what is actually happening. But in both of these methods, whether a correlation approach or this sort of hybrid mechanistic modeling approach, you run into cause-effect problems and stock flux problems. So we spent quite a bit of time in our group discussing some of those challenges.
Awesome. I love how the different groups went in fairly different directions. Great. So I guess with the last few minutes, I would ask, does anyone have any sort of comments about what they heard from the other groups, or comments speaking to the overall goal of this working group, which is to push towards new directions? Because, well, I'll see if anyone else has something to say, and if not, I can say something. I mean, I think a very sort of obvious direction that we're all pushing towards is understanding the functioning of mixotrophs in the water. So in terms of their biogeography, it's tempting, definitely, like Holly said, to talk about presence absence, but pushing towards tools to know what they're actually doing when they're present is critical. And I know we've talked about that over and over. I think that's like a very clear direction from this working group. Does anyone have any comments about that? Okay. I don't have anything further. If everyone is satisfied with that discussion, I think we can convene 5 minutes early. Is there anything to add from anybody? All right. I see no hands raised. Well, thank you so much for joining this meeting. It was really fun to discuss biogeography with you guys. Our next webinar meeting is when, Nicole? What?
Oh, sorry. It's in July, the second Monday in July, whatever date that is. I'm trying to pull it up. That would be July 11th, back at 11 a.m. The topic is remote sensing and mixotrophy.
Oh, great. So that dovetails really well with biogeography and is maybe a tool to push us towards at least a more synoptic view of mixotroph biogeography.
We'll see.
What?
I said, well, yes, we'll see. I sure hope so.
Okay, great. Well, thanks again, everybody, and I hope we'll see you at our next meeting. And tell all your Mixotroph enthusiast friends to join us next time. Take care. Thanks again for speaking, Kyle. Appreciate it.
Thank you,
Thanks a lot.
Kyle and Karen.
Thanks.
That was great talks, great discussions.
Thanks, Kyle and Karen.
Okay.
Bye bye.